Hi, welcome back to my YouTube channel. Today we'll be talking about a replication of telomere. What is a telomere? So, in case of eukaryotes, the telomeres are the end of the chromosome. So, because in case of eukaryotes, the individual chromosomes are linear, therefore you'll always find the end, and that end is a specific. It contains a specific structure and function, and that structure is called telomere. However, it is important to know that in case of prokaryotes, because most of the prokaryotes contain circular covalently closed genomes, therefore you will not find telomeres there. So telomeres are characteristics of eukaryotic chromosome. Now, one of the major problem that eukaryotic chromosome faces is that during replication there has to be a coordination between leading and lagging strand synthesis. Here is the replication procedure happening, and this strand is the leading strand. So leading strand is continuously synthesized from 5' to 3' direction, and lagging strand is synthesized discontinuously from 5' to 3' direction. However, apparently the movement of the lagging strand synthesis, the direction is opposite to the leading strand, but overall the replication machinery moves from this to that direction, 5' to 3' end. That means in order to continue the replication process, it has to form a loop on the lagging strand. So lagging strand is looped out so that the replication machinery it can move from this to that direction in an overall form. So a big problem that the eukaryotic chromosomes face is called the end replication problem. As the name suggests, this problem arises at the end of the chromosome. So this one is the continuously synthesized leading strand, and this one is the discontinuously synthesized lagging strand. So you know that because the eukaryotic chromosomes contain multiple origin of replication, so there will be one RNA primer which is elongated, which is basically extended by the eukaryotic DNA polymerase from 5' to 3' direction. So when this RNA primer is removed from the end, the gap is filled by the replication machinery because an existing 3' hydroxyl group is available. However, what happens at the lagging strand is that because there will be multiple RNA primers, those primers will be definitely removed. But at the end, at the end of the chromosome, when this primer is removed, what happens is that our replication machinery cannot add any more nucleotides here, which means this particular portion at the end of the chromosome remains so this portion remains blank, which means that there will be a single strand overhang at the end of the chromosome, so at the lagging strand. So that means in each and every cycle this lagging strand will be, one of the two strands will be shortened. That is what is called the end replication problem. As we are talking about the ends of the chromosomes, these ends are called telomeres in case of eukaryotic chromosomes. So these ends or telomeres are actually there to protect the chromosomal ends from being processed as a double-stranded break. So the cellular machinery cannot recognize it as a break. And this end protection relies on telomere specific DNA conformation, chromatin organization, and DNA binding proteins. Now I'll introduce you to another term called Hayflick limit. So in 1960, scientist Leonard Hayflick, so he discovered that in culture the human cells can actually grow a finite number of times, that is 50 generations, which he observed. So after that the cells will enter a senescence and ultimately it will die. So that means each and every cell will have its own lifespan. So this limit of lifespan is called Hayflick limit, and now you have understood that this Hayflick limit is because of the telomere shortening. Now interestingly, the end replication problem is a problem that is faced by the lagging strand. This difficulty is not observed during the duplication of the leading strand template, because during the duplication or replication of the leading strand template, a single internal RNA primer can direct the initiation of a DNA strand that can be extended to the extreme 5' terminus of its template, because it already has an existing 3' hydroxyl group that can be extended, right? The question is now how to solve this end replication problem, or if at all there is any solution. Yes, it does have a solution. In case of some viral DNA as well as some bacterial linear DNA, which is very rare, what happens is that this 3' hydroxyl group is provided by an existing protein. So the proteins will have the tyrosine amino acid residues, and that hydroxyl group of the tyrosine will actually provide the hydroxyl group, and that hydroxyl group can be used as an RNA primer which can be extended, and the telomere shortening can be prevented. Now if you ask me what happens in eukaryotes, yes, so the end of the eukaryotic chromosomes can actually solve this problem, because human telomeres, example of eukaryotes, consist of many head-to-tail repeats of the sequence TTA-GGG. So these repeats are, although double-stranded, the 3' end of each chromosome extends beyond the 5' end of the single-stranded DNA. Now what happens is that a unique enzyme is present in many cells called telomerase. So this telomerase helps extending the end of the chromosome in case of eukaryotes. Now let me talk about a little bit detail about this telomerase enzyme. So you should know that telomerase is an example of ribonucleoprotein. So as the name suggests, the ribonucleoprotein consists of protein as well as RNA component. That is why the name is ribonucleoprotein. Now you can ask, what is the function of this RNA component present in the telomerase enzyme? So this RNA component serves as the template for adding the telomeric sequence to the 3' terminus at the end of the chromosome. So basically this RNA component serves as the template. Although the size of this RNA component varies between 150 to 1300 bases, in case of human, the sequence is a repeat of AAU CCC AAU from 5' to 3' direction. So what is the function of this specific sequence? This region of the RNA can actually anneal to the single-stranded DNA of the telomere at the 3' end of telomere. And this annealing occurs in such a way that a part of the RNA template remains single-stranded. So this single-stranded portion can be actually creating a primer template junction that can be acted on by telomerase. So the bottom line is that the RNA component serves as the template that anneals to the single-stranded region of the telomere in such a way that DNA extension can be done. Now the question is, what is the function of the protein subunits? One of the protein subunits of the telomerase enzyme is a member of a class of DNA polymerase that use the RNA template. It is called reverse transcriptase. This subunit is called telomerase reverse transcriptase, or TERT or TERT. So it copies the telomeric RNA into DNA instead of the more conventional transcription of DNA into RNA, and this is opposite to the central dogma. That is why the enzyme is named reverse transcriptase. Now once the reverse transcription happens, at this point the RNA template disengages from the DNA product and re-anneal to the last 4 nucleotides of the telomere, and then repeats this process. So this process is repeated several times in such a way that the existing 3' end can be extended. So in this section you will find the difference between DNA polymerase and telomerase. For example, the telomerase contains an RNA component which DNA polymerase does not, and then a lack of the requirement for an exogenous template. That means telomerase already contains the template. It does not require an exogenous template. However, DNA polymerase does require an exogenous template. And 3, the ability to use an entirely single-stranded DNA substrate. So that is what is used by telomerase but not DNA polymerase. So, and the fourth and another interesting property that telomerase has is that it has a helicase activity. So the RNA DNA duplex can be opened up. That is what is called helicase activity, and DNA polymerase does not contain this helicase activity. Now there are also certain similarities between this DNA polymerase and telomerase. For example, both the DNA polymerase and the telomerase, they require a template, so they are template dependent to direct the nucleotide addition. That is the one. And second, they can only extend a 3' hydroxyl group at the end of the DNA. That means they cannot, both of them cannot initiate DNA synthesis de novo. And 3, both of them use the same nucleotide precursors. So the same nucleotide precursor pool is used from where? both DNA polymerase and telomerase perform this DNA synthesis, and they also acts in a processive manner, adding many sequence repeats each time it binds to a DNA substrate. Here's the picture now. So this is a telomerase enzyme. Here you see the telomeric RNA component. This anneals to the part of a single-stranded DNA. So there are 4 base pair annealing, and then this existing 3' hydroxyl group is extended by this telomerase enzyme machinery. So the telomerase uses its reverse transcription activity to synthesize DNA to the end of the RNA template. So this red portion is the DNA, and then the telomerase displaces the RNA from the DNA product and rebinds at the end of the 5' telomere. So here it rebinds, so it disengages from this location and then translocate to this particular location, right? And then this process repeats several times so that telomere can be extended considerable length of DNA. So this is in brief. So this red portion is the portion that is extended by the telomerase, and then you see now once the length is considerably long at the end of the telomere because of the action of the telomerase enzyme. Now what happens is that the DNA repair machinery, they come and they again here they synthesize the RNA primer, and then they add the complementary base pairs, right? That's the mechanism telomerase perform its action. Alright, that's great. You learned about the function of the telomerase today. If you think this video is useful to you, please hit the like button, subscribe my channel, and don't forget to put a comment in the comment box. Thank you. Bye bye.